Namaskar dear students, welcome you to our channel by Gulab Sir. As a part of series of lectures, today's topic is vacuole. We learn about endomembrane system. This is the last member of endomembrane system. Can you remember which organelles are the part of endomembrane system? Very first, endoplasmic reticulum, Golgi body, lysosome, and now this is the last one as a member of endomembrane system, vacuole. So in that connection we have to have this discussion. Whatever produced in endoplasmic reticulum, it is become a part of Golgi body. Whatever produced, modified and transported by Golgi body, it throughout the cell, remaining component content, that is what enzymes form the lysosome, as well as that content which are produced and they are extra, as well as cell absorbed from outside, all the components they get stored into the vacuole. So vacuole is also known as storage house of cell. It is also known as bag of the cells, where that all the components they are stored. I have made the list. Vacuole is a single membrane bound organelles. It has this membrane around it known as tonoplast. It is also made up of lipid and protein. It is also 40 to 60 angstrom in thickness. It is also selectively and semi-permeable as cell membrane is, and therefore which component and how much concentration and selection which supposed to be allowed to in and deposited. Very important thing to note about vacuole and tonoplast is that tonoplast help to absorb or content pass into the vacuole against the concentration gradient with the environment of energies to store them as they are extra, and therefore concentration of content present in the vacuole has more as compared to the cell sap. That means the medium of vacuole is hypertonic than that of medium of the cell sap cytoplasm, and that is very important to note down as a part of NEET. Once again, tonoplast allow to transfer the content against the concentration gradient, and therefore the concentration of component or sap present in the vacuole has more as compared to the cell cytoplasm. Now come to the point again. Here I have mentioned what I am saying to you. It is a single membrane bound organelles. Membrane is known as tonoplast. It is very large size in plant, almost cover 90% area of the plant cell. Why it is not large size in animal? Plants are stagnant, they are at a place. During favorable condition, whatever they get so, they do absorb and they store it in vacuole, because as and when they need, they can use from vacuole. But in animal cells, since they are mobile, they can go one place to another place, and therefore their cell need not to store the component and content. They usually get all the content and component time to time. Therefore, animal cells, they have smaller vacuoles and many vacuoles. So this is the difference between vacuoles of plant cell and animal cell. In young cell they are absent because young cell need not to store, that is under the growth, continuously dividing and dividing to produce the new cells. So newly produced cells, since they are just produced, therefore they do not have vacuole. As well as RBCs do not have vacuole. RBCs do not have other organelles also. So they store various components, and I have made this list. See, go through it. Organic acids, carbohydrate, lipids, nitrates, enzymes, amino acids, alkaloids, tannins, anthocyanin, water sap, excretory products and ions. Very important to note anthocyanin. Anthocyanin apart color to the flower, and I have mentioned in notes. See here, clearly mentioned. They store anthocyanin pigment which gives color to the flower. So question can be asked. During flowering period, this anthocyanin from vacuole transfer from leaves and stem to the part or floral bud which is growing on. So naturally this anthocyanin give an apart color to the flower. Very important that protist, amoeba and protist. Especially I have taken out amoeba from protist because we used to have often discussion about amoeba. Amoeba has various vacuoles called food vacuole, digestive vacuole, absorptive vacuole and excretory vacuole. So in amoeba, this vacuole not only help in digestion but also in excretion and overall osmoregulation of that cell, because amoeba single cell organism. So that single cell has to manage all, and vacuole play a vital role in digestion, excretion and osmoregulation. So all the protists, in all the protists, this food vacuole, they help in engulfing and digesting the food. Therefore, this vacuole as a part of digestive system. So please remember these two things, they are also very important. Now, once again I repeat that point which was I refer to you, that can be asked in NEET. Tonoplast facilitate the transport of ion against their concentration gradient, and hence the concentration of these ions is very high in vacuole as compared to the cell cytoplasm. That is the point of need to be remembered. Question can be asked. Also, vacuole maintain the cell pressure, turgor pressure of the cells, and therefore overall size of the plant cell is because of this vacuole. Vacuole also help to store various wastage material by the time which cannot be released by the cell. Therefore, not only store the important component but also it store the waste material, and that is what already I mentioned to you. Please go through this list so as to know about the component which are stored in vacuole. Question can be asked, what is tonoplast? Mention its role. Also, what are vacuoles? Mention the role of vacuole in cell. Why the plant cell vacuoles are larger in size. We mention the importance of vacuole in protist and amoeba. This can be asked. I hope you got it. This is very simple organelle, but remember at the same time this is a member of endomembrane system. Question asked in NEET, one of the following group of organelles, they are the part of endomembrane system. So remember 4: endoplasmic reticulum, Golgi body, lysosome and vacuole. I hope it is very clear to you. I am happy that you are always seeing our video. Thank you. Visit again.